Today we will discuss about one of the pathways that plays an important role in the growth of uh, the growth of the cells, that is Hedgehog signaling pathway. Hedgehog signaling pathways. So we will first understand the signaling pathways and how these hedgehog signaling pathways are being used by cancer cells for their uncontrolled growth rate, and what are the different ways that has been identified to prevent those hedgehog signaling pathways. So this in order to in order to efficiently work for these hedgehog signaling pathways, there is a precursor that is usually formed in the endoplasmic reticulum. That precursor is SSH precursor that has two parts, that is SSH-N and SSH-C. And this C terminals of this SSH-N is usually connected to the cholesterol, whereas N terminals of this SSH-N is connected to this HHAT. So, uh, however, when it comes to this SHHC, it usually gets degraded as soon as it gets out of the endoplasmic reticulum. So all functions are happen because of this SHHN precursor. So after it goes out of the endoplasmic reticulum, there are different ways by which this can secrete outside the cells. First one is through this DISP1, where this SSN will go out of the cells and can interact with this SCUB2. In other case, this SHHN can directly go out of the cells in the form of this multicellular, not multicellular, the we can say the combined SSN, or the poly SSN will forms, and then it can easily go out through the cells. In the next case is with the help of this HSPG receptors, where the lipoprotein can act as a vesicles that will load this SSN and can transport it for a long distance. And next, last one is with the help of this PTCH transmembrane receptors. It's a negative feedback receptors that helps in the formation of this precursor, that is SHHN, and helps them to transport out the cells. So this is about these Hedgehog signaling pathways. Now let us understand, since it is related to this maintaining the growth rate, so these things are being exploited by the cancer cells. And in order to avoid these Hedgehog signaling pathways in the cancer cells, different steps has been identified and are being targeted. For example, when this SHHN is not present, then it's a act as PTCH is one act as a negative feedback loop, negative feedback control. There, what it will does is it will first detach this SMO, and thereafter this SMO can upregulates the transcription factor, that is GL1, and this transcription factors which upregulates will go into the nucleus and upregulates the proteins, or upregulates the genes. Yeah, upregulates the genes that are essential for this Hedgehog signaling pathways. So in order to avoid this process, the one thing the different areas can be targeted. For example, this SMO, to prevent this PTCS to detach this SMO, and the other ways is that the activated the GLI transcription factors uh, should not be get activated with the help of this SUFU uh, reagent or drug. And therefore what happens is that inactivated GLI transcription factors will go into the DNA, and it won't upregulate the Hedgehog target gene. The other ways is that this SMO can be prevented. SMO can be prevented using this enzyme, such as this SMO will not make this transcription factors itself, that is GL1, GL2, GL3, and through this A2 AS to O2, and thereafter if there is no transcription factors formed, then the activated GLI will not go into the nucleus or and upregulates these Hedgehog target genes. So these are some of the areas that are being targeted to prevent the growth rate of the uncontrolled growth rate of the cancer cells. So I hope you got a basic idea about these hedgehog signaling pathways and how people are targeting those hedgehog signaling pathways to diagnose the cancer, and simultaneously how the cancers are using these hedgehog signaling pathways for their own benefit. And if you need to understand more about this detail, you can go through the references that has been provided in the description and also below the figures. That's all for this video. Thank you for your time.